In this video, we're going to be discussing in some detail the synthesis of peptidoglycan. Now, remember what peptidoglycan is. It is a polymer of protein and carbohydrate, thus the name, and it serves to protect bacterial cell walls. So it actually exists in the cell wall, and then it protects the bacteria from host immune defenses and also environmental factors. Okay. Now, in this image specifically, we're actually considering first a gram-negative bacteria. In gram-positive bacteria, it's going to work the same way, but just recall that if we're talking about gram-negative bacteria, remember that gram-negative bacteria actually have two membranes. Okay? They have an outer membrane, and they have an inner membrane as shown right here. Remember, they're actually very similar structurally to mitochondria. So down here, where you can see all of this initial synthesis, this is the cytoplasm of the bacterium. Okay? And between these two membranes right here, the outer and inner, we have the periplasm, or sometimes you'll refer to it as the periplasmic space. So if you're talking about gram-negative bacteria, initially the first half of the synthesis occurs in the cytoplasm, and then the last half of it occurs in the periplasm. If we're talking about gram-positive bacteria, they lack an outer membrane. They just have one membrane. So this would not actually be the periplasm in gram-positive bacteria. It would really just be extracellular space. But essentially the synthesis is going to be the same regardless of which class of bacteria we're talking about. And initially, we're going to divide the synthesis into four stages. The first stage is going to be to build N-acetylmuramic acid, or NAM. You've probably heard that peptidoglycan is repeating units of NAM, NAG, NAM, NAG, where NAG with a G is N-acetylglucosamine, and that's true. It turns out that NAG, N-acetylglucosamine, is really easy to make. In fact, the synthesis, of which I've shown in a separate video, is very short. So NAG is easy. The trick is building the NAM, the N-acetylmuramic acid. You can kind of see here, we start with this right here, and we have to go all the way up here in order to get a functional N-acetylmuramic acid. So that's a tougher thing to do than making the NAG. The second stage is we're going to build a pentapeptide sidechain. This is just a sidechain of linked amino acids, of which there are 5. The third stage is going to be to actually make the NAM-NAG glycosidic bond, and all these processes 1 through 3 occur in the cytoplasm. In stage 4, we're actually going to flip all of this into the periplasm. If we're in gram-negative bacteria, it's the periplasm. In gram-positives, it would be the extracellular space. Okay? But again, we're just referring to gram-negative bacteria here. So let's actually look at these stages, and then we'll go on to the next slide, or the next stage of the synthesis. We're going to start here with glucosamine 1-phosphate. Okay? You'll notice that a lot of these enzyme names, they're actually represented by their gene names. Since we're dealing with bacteria, this is very common. So GLMU, this enzyme is going to convert glucosamine 1-phosphate into N-acetylglucosamine 1-phosphate. Then GLMU is going to convert N-acetylglucosamine 1-phosphate into UDP, N-acetylglucosamine 1-phosphate. So we actually have to get this UDP on there because we actually have to activate the carbohydrate in order to attach it to other things later. All right. Now we have MURA, or MURA. This is going to convert UDP N-acetylglucosamine 1-phosphate into UDP enollyl pyruvyl N-acetylglucosamine. This is kind of a tongue twister here. And then we're going to have GLMU, which converts UDP enollyl pyruvyl N-acetylglucosamine into UDP N-acetylmuramic acid. Okay, so here's our N-acetylmuramic acid right here. We've actually generated it. We're actually going to have to do one more step, and it's actually going to be a hydroxylation of the acetyl group on N-acetylmuramic acid. Now, for obvious reasons here, because the sake of space and also complexity, I'm not showing you the structures. But you can actually take a look at this next structure up here, and you'll see that there's actually an OH group that's actually on the acetyl group. Okay, so we're going to convert UDP N-acetylmuramic acid into UDP N-acetylmuramic acid with this glyc. And this glyc just refers to the fact that there's that OH group on the acetyl group. All right? And this is catalyzed by NAMH. So that accomplishes stage 1. We've now built our NAM, N-acetylmuramic acid, in its mature form. The next step, stage 2, is to build the pentapeptide side chain. So this is going to occur with 5 enzymes, which makes sense because we're actually adding 5 different amino acids. So the first thing we actually have to do is we have to condense 2 molecules of D-alanine. Now notice this is not L-alanine. Generally, in pretty much every situation that we could have, and especially in eukaryotes, amino acids are in the L isomer form. We don't see D amino acids except for one, but that's an irrelevant point here. So in bacteria, this is one case where we see D amino acids. So we're going to condense 2 D-alanines right here, and this is going to be through an enzyme called DDL. Okay? Now, we're also going to have to add these other amino acids. So we're first going to attach the L-alanine, then we're going to add something called D-isoglutamate, and then we're going to attach a third one called mesodiaminopimelic acid, okay, or MDAB. These 3 are going to be added sequentially by μC, μD, and μE. Okay. As I mentioned up here, DDL condenses 2 molecules of D-alanine together, and then once we have this dipeptide, then μF is going to add this dipeptide onto the mesodiaminopimelic acid, and we see here the 2 D-alanines there. Okay, so that actually gets us our pentapeptide side chain. Okay, now what you'll notice up here in the membrane of the cell, it would be the inner membrane, of course, if it was the gram-negative bacteria. We have this molecule called bactoprenyl. So bactoprenyl is an important carrier of different molecules, and it actually has this phosphate right here. That's what this P is. What will happen is this bactoprenyl will actually get linked to this entire moiety that we've just created. So basically what this phosphate does is it attacks this phosphodiester bond that's present in the UDP moiety. And so it knocks off UMP, and it effectively links the bactoprenyl to this thing we just created. This is catalyzed by μX. So what we see now is we have a bactoprenyl in the membrane, 2 phosphates right here. This is a new phosphodiester linkage, and it's now linked to this whole thing right here. Okay, so hopefully that makes sense. Now, μG. What does μG do? Well, μG is actually what makes the NAM-NAG glycosidic bond, and it's just going to make 1 for now. So notice here it takes an N-acetylglucosamine and links it to the N-acetylmuramic acid. So we now have a NAM-NAG bond. Again, the NAG is really easy to make. Again, if you want to see how it's synthesized, you can go watch that in a separate video. But we've made our NAM-NAG bond, and that's stage 3. Now, all of this that I've just talked about, of course, occurs in the cytoplasm. And that's regardless of whether or not you're talking about gram-positive or gram-negative. It's the same. So if we're talking about gram-negative bacteria, stage 4 is going to be to flip all of this to the periplasm. We're going to flip this over into this space right here. If this were gram-positive bacteria, it would be flipping it into the extracellular space. This flip is catalyzed by a couple of enzymes right here, μJ and FTSW. They work together and they flip this up here. What do I mean by that? Well, now I flipped them up into the periplasmic space. All right. So that was what we just did. So what we just did is we flipped up this thing that was a NAM-NAG. But of course, peptidoglycan is a polymer, so it has to be NAM-NAG, NAM-NAG, NAM-NAG over and over and over and over again. So we're going to have to do transglycosylation, which is basically NAM-NAG chain extension. So this entire process that I just described, making all of this, and then flipping it up here, this happens over and over again to provide the monomers. This thing right here is basically the monomer. So for transglycosylation, we're just going to build a bunch of these, okay? And they're going to get linked together. That's the transglycosylation. Now, once we have transglycosylation, we're going to do something called cross-linking. Okay, that's actually what we're going to talk about on this slide. It's actually a process called transpeptidation, and that's actually the cross-linking, and then we're going to have some final processing. But in order to do that, we need 2 separate chains of peptidoglycan, at least 2. So here's one chain that we made, and here's a second chain. So as long as we have 2 separate chains, we can do these 3 things. And this is stage 6, the cross-linking and final processing. Okay, the first type of cross-linking we can have, I'm representing it by 2A here, that's transpeptidation, and I have this 3 to 4 here. What that means is that we're going to link the 3rd amino acid in the chain to the 4th amino acid in the other chain. So for example, mesodiaminopimulic acid is amino acid 3, but right here, this dealanine would be amino acid 4. And so if I link these two, that would be a transpeptidation, and it would be a 3 to 4 transpeptidation. So hopefully that makes sense. Here's another transpeptidation that's in a 3 to 4 fashion. Over here on the right, we're going to have a 3 to 3 transpeptidation. So in both of these, since all the side chains are the same, the third amino acid is mesodiaminopimulic acid. And so if we have a 3 to 3 transpeptidation, that means we're going to attach the mesodiaminopimulic acid here to mesodiaminopimulic acid here. And notice that on this one, since this is the one that actually is attacked, we actually lose the D-alanine dipeptide. Okay? That's a 3 to 3 transpeptidation. Now, the final transformation we can have is really more of a processing thing. This is not going to be a linkage between two chains. It's simply what's called a carboxypeptidation. And basically what happens is in both of these cases, we end up losing the terminal D-alanine. Notice that we've got this D-alanine up here linked to another D-alanine. In these chains right here, the terminal D-alanine is missing. It's actually been removed. And through the mechanism of this enzyme, it actually causes a carboxyl group to actually form where the bond was actually split. Okay? And so you'll actually see these things occur all throughout the peptidoglycan polymer. And that's how we get a mature peptidoglycan. And as we mentioned, going back to the first slide, if we have gram-negative bacteria, so let's actually put that membrane back up here. Remember that in gram-negative bacteria, we have this periplasmic space because there's two membranes, an outer and inner. And so that peptidoglycan, that is the mature form, will actually be in the periplasmic space. However, if we're a gram-positive bacteria and we don't have an outer membrane, it'll just be out here in the extracellular space. And the overall purpose of the peptidoglycan is to provide some resilience to the bacteria and protect it both from immune defenses, if it's in an organism like a host, or from environmental stressors. So hopefully this made sense to you. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.